Hi everyone, my name is Magda and I'm from Maynooth University under the supervision of Professor Kavanagh as part of Molecules 3. My project is on the evaluation of novel antimicrobial agents for use in treating drug-resistant bacterial and fungal pathogens. During the antibiotic era in the mid-1900s, we saw many medical advancements for the treatment of microbial infections. However, the emergence of resistant pathogens due to overuse and misuse of antibiotics have rendered many existing treatments inactive. The emergence of resistant microbial isolates will continue to rise and are estimated to cause 10 million deaths annually by 2050. We therefore urgently require new and/or repurposed therapies to help combat this issue. The aims of this work are to study a range of novel antimicrobial agents, to assess their efficacy, determine what pathogen they are most active against, and gain insight into their modes of action. In collaboration with Dr. Darren Griffith from Molecules 2, we have carried out work on the complex gallium maltolate, which has demonstrated good inhibitory activity against the gram-negative bacterium Pseudomonas aeruginosa. This is an opportunistic pathogen which poses a risk to immunocompromised individuals such as those with cystic fibrosis and has proven difficult to treat with the emergence of drug-resistant isolates. Gallium is a semi-metallic element with structural similarities to iron. Because of this, it gets incorporated into cells in place of iron, thereby disrupting iron-dependent processes such as DNA synthesis and stress defense as examples. However, the exact modes of action remained unclear. In vitro toxicity studies determined the efficacy of gallium maltolate and guided subsequent in vivo studies using larvae of the greater wax moth as an insect model. These larvae can help assess drug toxicity and also the protective ability of the drug against infection in vivo. The chosen test concentrations of gallium maltolate displayed no adverse toxic effects and maintained 100% larval survival following Pseudomonas infection after 24 hours. Our ultimate aim of this work, however, is to gain a deeper understanding into the modes of action of gallium maltolate through label-free quantitative mass spectrometry. Here we can treat the bacterial cells with gallium maltolate, extract all the proteins, and analyze protein abundance changes in response to the treatment to deduce potential drug targets. With the resulting mass spectrometry data, we carried out graphical and statistical analyses. The heat map on the left shows the distinct contrast between gallium maltolate treated Pseudomonas aeruginosa versus untreated control samples. Proteins coloured red are those increased in abundance, whereas those in green are decreased in abundance in response to the treatment. The distribution of all identified proteins can be displayed on this volcano plot above, where we can look at specific proteins of interest. Proteins on the right-hand side are those elevated in response to the treatment, whereas those on the left have decreased. Proteins associated with oxidative stress, DNA damage repair, and iron acquisition are among those elevated in treated samples, whereas proteins required for virulence, flagellar motility, and quorum sensing were decreased in treated samples. This is also highlighted in the string network analysis below, giving us insight into the potential compensatory mechanisms for inadequate iron supplies within the cell, and also alternative methods in which the complex is inhibiting growth. In addition to previous reports, this provides more insight into the modes of action of gallium maltolate, which in turn may also guide and enhance drug synthesis. Gallium maltolate initially came into use as an anti-cancer agent, but with the onset of antimicrobial resistance, it has been repurposed as an antimicrobial agent. Our work has highlighted the potential of this drug in treating Pseudomonas infection, both in vitro and in vivo. Toxicity evaluation in wax moth larvae have shown promising results, which are aimed to subsequently inform mammalian toxicity studies. A big part of our work is proteomic analysis, which is a useful tool to better understand the effect of a drug at a molecular level, which in turn can help inform drug synthesis to create a better formulation with enhanced activity. To finish off, I'd like to thank my supervisor, Professor Kavanagh, to our collaborator, Dr. Darren Griffith, to SSPC for funding our work, and to you all for listening. Thank you.